Hello, everyone. Good morning or good afternoon, depending on your time zone, and welcome. Today, we are discussing how pupil diameter can be used in conjunction with eye tracking, including some best practices and showcasing a tool that can help with preprocessing and analysis. This webinar will be recorded, so feel free to rewatch or share with students and colleagues. A link to the webinar recording will be sent via email within a few days. And we've also hosted many webinars like this in the past, which can be found on our Tobii Resource Center. This is our agenda for today. So first, I'm going to quickly share an introduction to pupil diameter metrics and our Tobii products. We will have 3 presentations. Dr. Mariska Kret from Leiden University will be first, followed by Anouschka van Dijk, also from Leiden University. And our final presenter will be Dr. Sylvain Sirois from the Université of Québec, Trois-Rivières. At the end, we will have our Q&A roundtable discussion. So please feel free to ask questions in the question box throughout the presentation, and we will address them at the end. So why do people use eye tracking? It is an objective and quantitative way to understand human behavior. It allows us to capture data from participants without any subjective or conscious filtering. Eye tracking provides insights into the cognitive processes underlying visual behavior and attention. Eye tracking, especially by Tobii, is unobtrusive and non-invasive. We do not require chin rest or head rest to use our systems. They can plug into a computer and blend right in. Tobii eye tracking systems are versatile, portable, and easy to use. You can collect eye tracking data anywhere, anytime. We have systems even as small as to fit into a backpack. And eye tracking works together with physiological or neuroscientific measures that you may already be using. It's a great addition to data streams that you may have. Tobii eye tracking also provides a variety of eye movement metrics like fixation, sense of cons that a lot of people are aware of, but we also provide eye-related biometrics like blink data and pupil diameter estimates, which is what we're going to be talking more about today. The pupil is an important part of how Tobii eye trackers work, as we use a technology called pupil-centered corneal reflection. So our cameras take images of the human eye and find the center of the pupil, and the position of the corneal reflections calculate the participant's gaze. The pupil is also an integral part of the human eye, as it's the opening at the center of the iris and allows light to pass through it to the retina. It's a light-sensitive muscle of the eye. It's not possible to exert control over this pupillary muscles. The iris adjusts the size of the pupil to allow more or less light. But even at a resting state or under constant lighting, researchers have found the pupillary changes in response to a variety of situations. Pupillary muscles receive input from brain systems involved in cognitive and autonomic functions. Here are some examples of how pupil diameter changes can inform your research. Pupil diameter changes have been related to attention and attentional networks, memory, like memory strength or recognition, decision-making, reward value, effort, or uncertainty, associative learning, fear conditioning, and Pavlovian conditioning. Pupil diameter changes have been related to cognitive load, like information processing and increased mental overload, and even affective systems, so increased emotional arousal is related to increase in pupil diameter. Task-evoked pupil dilation is connected to underlying neural systems of the brain, which indicates a relationship between cognition and pupil dilation. So being able to capitalize on this connection allows researchers to make inferences about neural systems in cognitive studies. This is an overview of our Tobii Pro scientific products. All of our eye trackers provide estimates of pupil diameter for left and right eye separately at the sampling rate of the system. We have several dual camera systems that tend to be more stable for measuring changes in pupil diameter over time. The Tobii Pro Spectrum and Tobii Pro Fusion are our recommended options for screen-based systems to measure pupil diameter changes. You can also get pupil diameter estimates for left and right eye from the Tobii Pro Glasses 3. For anyone who's watching who has an older eye tracker, feel free to reach out to us for guidance, but the Tobii Pro Glasses 2 will work with pupil diameter measurements, as well as the X260 and the TX300. If you have a system that you're curious about, just send us an email. Tobii Pro Lab is our software tool that provides all of these metrics included, and they all work with the eye tracker shown on the previous slide. You get gaze-related data and fixation synsicades, but also eye-related biometrics, both in visualization form and in metrics exports. So the pupil diameter measurements all come in millimeters. The average pupil diameter that you see for AOIs and intervals are calculated using the pupil diameter filter after data collection. And you also get all the raw measurement for each sample left and right. And the raw pupil diameter is in time series format, can be exported for use with other pre-processing scripts. And we're going to learn more about that today. So now on to our presenters. First, we have Dr. Mariska Kret, and she is here to talk more about her research. Welcome and take it away.
Kido. Yes, thank you. Hello, everyone. Thanks for being there. Yeah, so today I'm going to talk a little bit about pupil size in humans and also in non-human primates. So let me just go to presenter mode. Yeah. All right. So I'm going to start with the first part, give you an overview of pupil size, and then Anouschka will take over and talk about the pre-processing steps and the analysis part. So obviously humans are not the only ones that have pupils. So a lot of animals have them. And it's interesting to see the different types of shape that evolved in those different species. So I don't know if you knew this, but if you, for example, look at prey animals like goat or sheep, for example, you can see that they have horizontal slit-shaped pupil sizes. Whereas if you look at animals like crocodiles or cats, so predator species that live close to the ground, they have eye slits. And then predators that are higher up from the ground, they have more rounded pupils, like we do. So yeah, different evolutionary pressures selected for these different shapes. And of course, this has direct perceptual benefits. For a goat to have the pupils like that, it will give him a wider view of the environment and easier to detect predators. Okay. The pupil is controlled by two different muscles, the sphincter and the dilator muscle, and together they organize the contraction or the dilation of the pupil size. I have a little video that demonstrates that. So you can see the pupil dilator, the radial muscle around it, and then also the sphincter, the circular one. You can see that it reacts in response to light. So if it turns dark, the dilator muscle contracts. If it gets light, then the sphincter muscle contracts. So those two pupil muscles work together to let the pupil react to light, so that not too much comes in when it's very bright outside. Yeah, let me know if it doesn't change again. Then I will stop share and restart sharing. I don't know why. So here you can see the different images of those different species. Here you have the crocodile living close to the ground, having the slit vertical pupil sizes, and here a goat with horizontal pupil sizes, and us with the round pupil sizes. Then here is this video showing how they work together, those two different muscles. Can you now see this video?
Yes.
Okay, great. And then it gets dark and the pupil dilator muscle contracts, enlarging or dilating pupil size. And when it gets bright, the pupil contracts so that not too much comes into the eye and damage the tissue. So then the pupil dilator muscle is actually the pupil size dilates when people get aroused. So it's via this superior cervical ganglion cells, and it works together with norepinephrine. This is elicited by the locus coeruleus, whereas the sphincter pupil muscle contraction or decreases pupil size. It's more parasympathetic, so it's more during rest, let's say. So here you can see the sympathetic one. So when the pupil size dilates, you can see what else happens in the body. So your heart may start beating faster. So it's the bladder is inhibited. There's lots of other processes going on. Stimulating orgasms, another one. So the pupil dilates, and it's a measure of sympathetic arousal. And you can also see that because it correlates very highly with skin conductance changes. So skin conductance in humans, it's typically measured by electrodes on the fingertips, and it basically measures how much people are sweating. And you can see that in response to high arousal, pleasant, or high arousal, unpleasant images, the pupil size dilates and skin conductance increases. So you can see this really nice U-shaped pattern here. So please note there is no effect of valence here. So it doesn't matter whether the arousal is of positive or negative nature, the pupil size will dilate and skin conductance will increase. All right, so the measure of pupil size has become much more popular over the years. There's really an increase in the number of publications, and also the past couple of years, this has even increased further. So there's a lot of interest in measures of arousal, and pupil size is quite easy to measure thanks to products like Tobii, which I'm using a lot, which I'm using a lot in humans, but also in humans closely living relatives, great apes, so chimpanzees, bonobos, orangutans. We are doing eye tracking on. And there is some literature, mostly from monkeys, so the ones with a tail, those are not apes, linking pupil dilation to, similar to what Marisa just mentioned, anticipated reward, cognitive workload, task conflict, decision confidence, stimulus saliency. So there is some work on it in monkeys, and now there's also some studies coming out where researchers look at the pupil size in great apes. So here is a comparative study where you can see the pupil size reaction to unexpected sounds in monkeys and also in humans. So you can see the different colors. Pink is pink noise, red is monkey crew, green is whistle, blue frequency defiant, and black is the standard. So black is standard, and then when this sound is played, you see that the pupil reacts in about a second later. Okay. Now, actually, pupils are not just to see, but they're also to be seen. So humans are making a lot of eye contact, and we are already doing that from when we're born. Actually, it's an innate response to look at the eyes of another person. Those pupils stand out from colored irises if you have light eyes. And if your eyes are light enough for the pupils to be visible, then there's research showing that this actually matters. So there is research showing that we have positive associations with people with large pupils and more negative associations with people who have smaller pupil sizes. So we tend to like, trust, feel attracted to people with large pupils much more than those with smaller pupil sizes. So I found it really interesting. I was wondering why that is the case. And then by studying that literature, I came across a very old study by Eckhart Hess, who has done a lot of eye tracking research and pupillometry research in the 50s. And he almost anecdotally wrote about a report on pupil mimicry. It was in a paper in the Scientific American. And I was reading that and I was really intrigued. Do people mimic pupil size? And at that time, I was working in Japan in an institute where they have chimpanzees. I was working as a psychologist for the first time in my life with chimpanzees. And I noticed that they have beautiful eyes and they have beautiful pupils. And I wanted to know, does pupil mimicry happen in humans? Is that something that really exists? And also, if so, does our closest living relative, the chimpanzee, also mimic the pupil size of conspecifics? So in Japan, they already had a Tobii system. You can see it here. I don't know the type, but it's a very old one, as you can see. Um, what we did was we tested chimpanzees and humans in the same place, and we let them observe the eye region of humans and chimpanzees. So both species watching both species. And what we did with the eyes on the screen is we manipulated the pupil size. So we let the pupil size grow bigger, dilate, or constrict. And then with the Tobii, we measured whether the pupil size of the participants followed that. And we did find that pupil mimicry happens, and also that it was especially strong within species, humans watching humans, chimps watching chimps. Well, now we've conducted a lot of different studies in humans showing that when people mimic on the level of pupil size, they also have a tendency to be more trustful towards each other, to cooperate better. We have a lot of studies that, yeah, that this synchronization in pupil size or pupil mimicry, however you want to call it, is linked to social decisions, such as to trust someone. So right now we're also extending this research in dogs. We're just testing dogs at people's homes, and we have some preliminary data showing that dogs also mimic the pupil sizes, mostly of dogs, and also a little bit of humans. We're still hoping to collect data in bonobos. So this is stimuli, dog watching bonobo, dog in humans. So this is work in progress. All right, so I hope I gave you a little bit of an overview of the pupil, and now Anouschka will continue and tell you more about the pre-processing steps and the analysis part. So thank you very much for listening.
Great. Thank you, Dr. Kret. Thank you, everybody else. I'm going to change the presenter to Anouschka now. And excited to hear more. Thank you, everyone, for your questions so far and all of your engagements and thumbs up. Keep those questions coming. Is anybody, and we're going to ask.
Yeah, indeed. If anyone has a question for me, then I'm happy to take it.
Does eye color affect the measure of pupil dilation?
No, eye color does not measure or affect the measure of pupil dilation. So we had a lot of people with different eye colors, so in Japan, but also in the Netherlands and other countries, and we never found an effect of the eye color.
Sylvain, if you're ready, we can move on to your presentation, and then maybe we can come back to Anouschka. So
yeah, if that helps, I don't mind at all. Obviously, it changes the flow, but
yeah.
Well, okay. Well, I just made you the presenter, and we can see if Anouschka can work in the background. Thank you so much. Thanks, everybody.
Right. So I assume you can hear me.
Yes.
Very good. And you can see my slide.
Yes, we can.
All right. Do shout out if there are further problems along the way. But looking forward to Anouschka, who's now back, Can we hear you? No. Oh dear. Oh, hopefully you can work on that in the background while I'll do my bit. Thanks to the organizers for having me. I will talk about analysis of pupil data from a functional point of view, where we assume that the expression of pupil over time can be described by functions, and we can do the analysis that we're interested in on functions rather than raw data. So the outline of this talk, I'll give you a little background as to why I ended up using pupillometry and why it might be good for you. I'll talk about splines, which is the family of functions I use to carry out analysis. This has some bearing on the experimental design for data collection. Then I'll use an example of an infant study where we analyzed pupil data that way. This is a broad audience, I presume, so I'll have some adult data as well to showcase the same principles. And at the end of my talk, I've asked the organizers to share with you a bunch of links, including to some data and some code you can use to carry out these analyses if you want to learn how to use them or figure out exactly how they do the things they do. So in terms of background, I'm primarily a developmental person. I ended up doing infant studies by some fluke of how academic careers go. And in a typical infant study, you show infants, for example, situations such as these, either using actual stimuli or recorded videos and so forth. And in this case, there's a hand disembodied that went to make contact with a ball. What you would do typically is show this event several times to infants. That's called familiarization. Or if you're hardcore, you would habituate them. You use the initial measure of interest of the infants, and you keep repeating until their interest has decreased by some standard. And when you do that, then you can move on to a test phase where you'll show them different events, and you're interested in how much attention they pay to the different stimuli. So in this case, for example, the hand goes again for the ball, which is in a new location. That would be one type of test event. And in this particular case, the hand goes to the same location that infants were familiarized with, but in order to reach a different toy. Tasks such as these are used with infants because they're the worst human participants in psychology research, but they are interested in things, and varying levels of interests are used to make assumptions about what and how they represent information. Now, I've had an issue whereby in the infancy field, looking time, cumulative looking time, is typically used to assess, information processing in infants. So you have as a reference how much time they spend looking at something that's your reference, so something expected, and then you compare that typically with more looking to unexpected events. The question is, more looking, what does it mean? Is there the same processing as in the reference situation, and on top of that, there's extra processing, which is your discovery? That's typically how these experiments are discussed, but the point I'd like to make, if we blend these two colors, maybe we're looking at something entirely different with respect to how infants process unexpected, unlikely, novel events. So there's a suggestion that to a degree, a lot of comparisons that have been made to generate our knowledge and understanding of infants have been comparing things that don't quite compare. So pupillometry has been around for a while, since the 60s, going back to Hess and Polt's seminal study using pupil diameter as a marker of attention and arousal. And in the case of infancy, eye trackers have been in infant labs for quite some time, but until recently, as much as pupil information is in the data dump that many researchers had access to, was pretty much ignored. But over the past 16 years, it's emerged as a attractive alternative to looking times as an index of information processing in infant studies. And particularly, and this is a point me and colleagues have been making, it is sensitive to the temporal dynamics of information processing, which is incredibly useful when you try and figure out whether and when infants react to particular things in order to make assumptions about how they process information. I want to point out that in the 60s with Hess and Polt, there was at the time sort of a peak initial interest in pupillometry, and Hiram Fitzgerald was a pioneer of trying to apply these methods to infant studies. At the time, however, it was using infrared film sampling at 2 Hertz. Had to be shipped Kodak for processing, sent back to the lab days, weeks later, and then it was assessed by hand, where you would project the film on a screen and use a ruler to measure pupil diameter. Exciting, it works well as a measure, as Fitzgerald reported in the late 60s, but the process of using pupillometry is so tedious that it didn't really catch on until modern eye trackers, such as the ones that Marisa introduced, do it automatically for you, which is great. So you have the data, and again, working with infants, worst participants you can imagine. If you want some physiological measure of information processing, any electrode you connect to infants increases the risk that the infant decides that data acquisition is not for them. So non-invasive methods like eye tracking are really good. So I've been using splines, and specifically a type of spline called B-spline for basis splines to transform the raw data into functions to be analyzed. Because when you look at pupillometry data, it does look like something is expressed over time. In this case, this is data from a simple maths task where people had to out loud add 2 large numbers. Some problems were easy, some problems were hard, and this is what you get because the pupil is very good at picking up cognitive load and associated arousal. Like event-related potentials and EEGs, unfortunately, people faced with temporal data like that tend to create temporal bins, where you look at the difference between 2 conditions in the average pupil diameter at different times, and what do you do with that? Well, you typically would do like ANOVA, looking at the effect of problem difficulty, time, position, and the interaction between both, and you'd be like, oh, well, there's a linear component here for easy problems, and the hard problems, there's a quadratic and linear component, so they are distinct in terms of information processing. Very exciting. However, what you do when you do that is lose a lot of useful data. The previous slide was based on 8 participants who did 6 trials for 10 seconds, so sampling at 60 Hertz. We had over 28,000 data points, which with the temporal bins we reduced to 80 data points, which is quite sad. And there's the question of how many bins do you use and where do you put them? Well, arbitrary decisions might make it likely that you miss out on interesting information, so a decrease in statistical power. Whereas if it's data-driven, you look at when things happen, you increase the risk of type 1 error by analyzing what appears to be interesting after examining the data. And yet, the raw data expressed over time looks like a function of time. And the question is, which one? Would you fit a straight line? Would you fit a curve? Well, B-splines will do it very neatly for you. So you have essentially a collection of simple cubic functions that are linearly combined to approximate your original raw data. And what you do is essentially create a basis of splines with enough underlying functions to capture the key features of the raw data, but smooth out noise. And it is okay to actually look at data to see how well a spline fits individual recording before you actually process everything. And then you transform all your raw data into functions using that common basis. And now all trials for all participants are functions. And the only difference between trial and participants is the parameters, the same basis function. And you can do your analysis on those functions, which is great, because the result of your t-test or your analysis of variant is going to be a function itself, which is expressed over time. So is something significant? Fair enough. When is it significant? Even better. As far as how you go about collecting data for this type of analysis, basically, this is the basic, partly covered by Mariska. You want your testing room to be reasonably lit, and this is a poorly defined suggestion, but you do want your participants to, without any stimulation, to have, relaxed but not fully dilated pupils if you want to measure useful changes. You want to set up your participants to minimize data drops. In the case of infants, I like to put them in car seats close to the stimuli and eye tracker. Minimize movement. They're comfortable, less likely to get fussy and drop out. Adult participants, well, eye trackers such as Tobii accommodate some head movement, but a good chair that keeps people steady will help you a lot. Now, pupillometry as a function of different test events would require ideally that the luminance of all the stimuli is equal, so that your differences are not due to differing stimuli in terms of luminance affecting the constriction based on the brightness of stimuli. That could be difficult, but failing that, if you're able to use stimuli whereby a stimulus can be in category A or B, depending on how you assign participants, you can actually average out the noise from varying levels of luminance from your stimuli. And also, you want to prepare your stimuli and events in such a way that crucial moments, because you're looking at the expression of pupil over time, do not coincide with the pupil-like reflex. So at the beginning of showing a stimulus is the worst possible time to try and look for pupil responses. You need to let the eye relax a little bit from the onset of stimulation. The last thing you want to do, unfortunately, is test your eye tracker before your very first participant to see if you have systematic errors in pupil size estimation as a function of gaze position. The pupil is measured by cameras with an eye tracker that are fixed location, but as your eyes move, the width of the pupil or the height of the pupil, depending where you look, will be different. And as much as you may hope so, you need to make sure that your eye tracker does not systematically overestimate or underestimate the size of the pupil at different locations on the screen or the environment if you're doing environment-based eye tracking. For spline analysis, you need trials that have the same fixed duration for all trials and all participants. Oops, too quick. Failing that, if you use tasks where participants are somewhat free to look at stimuli and you're going to be interested when they look at a particular stimulus and you're going to sort of create an event or an area of interest trigger, you want to make sure that you have enough recording post the participant looking at the key item of interest so that you can cut all your recordings and align them relative to this key event. An example will help because this is all quite abstract at the moment. I was fortunate to be responsible for the logistics of this project, which was a multi-lab approach to analyzing pupil data from an infant study. We invited 6 other labs to take the raw data and carry out the analysis as they see fit with their own methods, trying to test for the same research hypothesis. I'm going to talk about the method I use, and you should feel free and encouraged to actually consult the paper to look at how other methods deal with pupillometry. So this is a task where a hand is going to one of 2 toys and the location of toys is swapped between trials. There are 4 different videos, and in this study, a particular infant, one of the 4 videos was chosen as the habituation stimulus. and presented repeatedly. So in this case, for example, an infant repeatedly saw the hand go for the ball, which was on the right, and then we tested infants after habituation on all 4 videos, the hand going for either toy at either location. The question is, does the identity of the toy have a bearing on the reaction of the infant? Does the location or path of hand movement have bearing? These are important to figure out whether or not infants attribute goals to human agents. Well, this is the average pupil response to the 4 different test events, and they are different events for subsets of children, but for each child there was a toy that was familiar, and there was a path that was familiar, and likewise there was a novel toy and a novel path relative to habituation. And what we see quite distinct from the others is this particular curve here, where if the hand takes a novel path to go to the familiar toy, that seems to elicit more responding from infants. Now, this is the average of raw data that's merely been interpolated to fill in gaps from blinks and head turns and smoothed using low-pass filtering. This is the same data averages from the splines that we use to capture the raw data. So I'll go back and forth a little bit. Hopefully it's not too dizzying for you. But what you see going from averages of raw data to averages of splines is just a little more smoothing, but the key characteristics of all 4 averages are maintained in the spline space. What we can do with a spline is carry out the functional F-test, and in this case, it's the interaction we're looking at, and we find a significant interaction between the start of the hand movement and the hand reaching the toy. If you have a significant interaction, you should be looking at simple effects rather than main effects. And if we're looking at simple effects of target, whether they react more to the novel relative to the familiar stimulus for each path, we find a significant effect of toy, more responding to the familiar stimulus during movement, and also sustained again after the hand has rested on the target toy. Simple explanation of otherwise ambiguous looking times, which I'm not presenting today, but that you'll find in the paper. The dynamics of events we show infants matter. Learning during familiarization or habituation matters in terms of the responding you see from infants in test trials, and experimental design matters as well. If you're interested in other ways to look at pupillometry, again, feel free to look at the paper. And the main drive for carrying out this project was to figure out whether and why there would be convergence or divergence between approaches. Conclusions in the paper. If we look at adult data, which may be of more interest to a majority of people attending, here's an example of the gaze data from a task I'll talk about. Simple task, fixation cross appears in the middle at the beginning of a trial. A circle appears left or right, and when participants shift their gaze to the circle, there's a halo that appears around the circle and a sound is played. A simple task. This is data from 20 people. The blue dot is the average of all participants, and you see that it seems to be pulled towards the upper left. That's because of all the missing data that have not filtered out, coded as -1. So when people blink, there's a -1 in the stream of data, and the average just reflects that bias. Now, in this particular task, 20 young adults taking part in a summer course on neuroscience method, including pupillometry. It's an auditory oddball, 64 trials, 56 of which had a standard sound, which hopefully you'll hear. But 8 of the trials had a auditory oddball, 4 of those using a low frequency sound, and 4 of those using a high frequency sound. What does it look like? Well, if you need data that's super clean to illustrate the property of the pupil, use an auditory oddball, because this looks made up, but it's from 20 participants, and every year that I teach the course, it looks like that. It's extremely reliable. So you have the change relative to baseline, the diameter of the pupil at the onset of the sound, averaged for the standard sound, the most frequent one here, and both oddballs, the solid line, with slightly different profiles for the high frequency relative to the low frequency oddball. And if you do a t-test on the functional data derived from the raw data, you find that essentially about half a second after sound onset, there's a significant increase in pupil diameter for auditory oddballs relative to a standard. Tells you what obviously the averages tell you, but tells you also when, in a way that you don't have to arbitrarily decide, which I find quite attractive. So this is a quick overview. I'm sorry if it's overwhelming. You will be provided with links to the finer details of this approach, but I do like functions because they're extremely flexible. They're quite powerful at capturing data and allow analysis that make use of all the data, telling you whether, but also when things are worthy of your interest, without making arbitrary or data-driven decisions about when and how many temporal bins you should apply to analyze your data. Some constraints: you need durations identical between trials and between participants. That can be tricky for some experimental situations, but all the same, make the decision a priori, and you can have very fancy analysis tools done relatively easily. You do need some programming fluency. That can be scary, but one advantage is if you go through the learning curve, you know exactly what you're doing to the data, and that's a very comfortable place to be when you want to talk about your findings. And as much as I use MATLAB and the code I share has been written in MATLAB, you don't need MATLAB because my scripts are text files, so are my functions. So with computer literacy, you could figure out and duplicate my code in other languages. And kind people such as Sam Forbes from Durham University has ported many pupillometry methods, including the ones I've been presenting today, for the R package, which is open source and freely available. So here's a link there for you. Love, Sam. Thank you very much.
Thank you so much, Dr. Sirois. Thank you for all the questions that have been coming in the chat. I'm really excited to get to those later, but we are going to try once more to have Anouschka present. Thank you, everybody, for your patience, and thank you, Anouschka, for working so hard to present. Super excited to hear about this.
Thank you so much. I'm so sorry for all the errors. I tried about 4 headphones, a separate microphone. I'm not sure what the issue is, but you can see my screen, I think, right now.
Yes.
So I'm going to be talking about the pupil dilation pre-processing steps. So I am a PhD student from Mariska Kret's lab, the comparative psychology and affective neuroscience, also in Leiden. And from Mariska, we heard that it can be used as an index for arousal and it's a non-invasive measure. And that's very good for, in particular, non-human primate research. So I will be showing you some pre-processing steps using the PhysioData Toolbox. And in the second half, I will be showing you kind of a demo of how the PhysioData Toolbox would work. So most eye trackers provide users with pupil size, usually a left pupil and a right pupil, but pupils do require further pre-processing. So usually eye trackers do give you with a valid sample and an invalid sample of your points of their pupil size, but there is some further pre-processing that needs to happen. And Mariska, Kret, and Anouschka, they created this pre-processing pupil size guidelines and codes. And based on that code, Anouschka later created the PhysioData Toolbox with a user-friendly interface where you can also visualize that data. So the first step would be to prepare the raw data, and the only preparation the PhysioData Toolbox needs is really to filter out all your invalid data samples. And then you can import your files into the PhysioData Toolbox. They give really nice visual overviews, so you can do your manual inspection of the samples as you go. And again, these are based on those guidelines of pupil pre-processing. PhysioData Toolbox is free to use for everyone. And I will be going through the data of this orangutan study by a former colleague, Tom Roth. It's a preferential looking task. So basically they have one orangutan with cheek pads on one side and a male without cheek pads on the other side. And they looked at how female orangutans respond to these males with cheek pads. So each trial consists of a calibration, then a fixation. Fixation is of their caretaker, Frank, for 1 second. Then they see the stimuli, so 2 orangutans on either side. And we expect a bigger pupil size for the attractive male orangutans with cheek pads, as cheek pads are a sign of an attractive orangutan male. So this is an example of what the task would look like. So this is the calibration. On the top right, you can see one of the female orangutans doing this task. You can see on the left side the male with cheek pads, the right side the male without. And you can also see the gaze point, and this is caretaker Frank as a fixation in the meantime. You can see, I don't know if you can see it clearly, but the orangutan is drinking from a nozzle, and it has very slightly sugary water in it. That is so we can kind of have a stable head position so the eyes can be captured accurately. Okay. So once you have collected your raw data and you've discarded all your invalid samples, you can put your data into the PhysioData Toolbox. So officially, the PhysioData Toolbox can only read PhysioData type files, but there are converters already in the toolbox. So for example, there's a converter for the ePrime extension for Tobii. So ePrime is a software where we can easily create these simple preferential looking tests. But also other type of data files are also you can also use, and there's a separate MATLAB script also free to convert other file types. And if your file type is not on that, you can also contact the creator, Edio Shakshi, and he can maybe make the another convert MATLAB script for the other file types. Okay, so you can use the file converter for this, and this is kind of what it would look like. You can have all your signals that you need that can be converted into the files for a PhysioData file. And once you've done that, you can open the session manager. So in the session manager, you can see, you can manually inspect the quality of the data. So as you can see here, I have an example of pretty clean data and very, very noisy data. So overall, you can see that these data points for the top line, it should kind of look like this, more of a stable, clear line. And on the lower side, you can see that one of the pupils, so the orange line stands for the right pupil and the blue line stands for the left pupil. And you can see they captured the right pupil pretty accurately, but the left pupil is very, very noisy. So you can see these deviations in pupil diameter that are fluctuating very strong between 2 and 4 within milliseconds. So you can see, okay, this is probably not accurate data. And you can also already look for artifacts. So we have different types of artifacts. So, you know, something is probably an artifact if it is outside of these ranges. So the range, physical range of a pupil for humans is between 1.5 and 9 millimeters. So if it's outside of these bounds, you probably already know, okay, this is not an accurate data point. And other artifacts can occur when participants blink. So when participants blink, especially halfway during the blink, the pupil is partially occluded. And sometimes the eye tracker does capture that as a very small pupil diameter. So that can result in different types of artifacts. So the PhysioData Toolbox is aimed at identifying these artifacts, not only for blinks, but also other parts. But manual inspection is still necessary, as you'll see later. So this is an example. You can see this small island here. That is a clear deviation from the trend line. So this ideally should be captured by the PhysioData Toolbox filters and filter this out as invalid samples. So here also you see this isolated sample. You see probably a blink or the gaze is redirected temporarily. And in the middle, you kind of see these vaguely sparsed like data samples. And these would also be captured as invalid. And lastly, this is something that you can really see clearly with a blink. You can see on the right side, you can see this very strong increase in pupil size. And this is kind of an naturally quick increase. And we can see, okay, it is right before data was not captured for a little bit. So we suspect that this was a blink. And you can really see that the pupil was already captured by the eye tracker when it was partially occluded during the blink. So this would ideally also be captured by the PhysioData Toolbox filters. So the PhysioData Toolbox, they create a mean pupil size that is the mean from both the left pupil and the right pupil. It will do smoothing, so there's a low pass filter to create a smooth continuous signal. As with pupil and other types of signals, the raw data is naturally quite noisy, and this filter will ensure like a smooth continuous signal. It will also upsample the sampling rate to 1000 Hertz. And it can also do interpolation. So small gaps, they can interpolate this mean pupil size through. But for large gaps, it will be set to missing. What is a large gap? It's naturally set to 250 milliseconds, but you can make that smaller or bigger depending on your preferences. But all the default settings in the PhysioData Toolbox are based on the pre-processing guidelines. So this is what it would look like in the PhysioData Toolbox. I'm going to be showing you that in a little bit, but just as a quick overview, you click on Create a New Analyzer, and here in the settings on the right, you can see all of the different filters that it will apply. So you have these isolated samples, which are these islands that clearly deviate from the trend line. You have the dilation speed filter and the deviation filter, and those are both if you have a few samples that are very far out of the normal trend line, then they will be discarded. You have the smoothing and the interpolation, and you have the edge removal filter, and that's the one that's specifically for blinks. So once you've applied all the filters, this is the output that you will get. So this is output of a very noisy signal. So this is with all the applied filters, and you can see it creates a mean pupil signal, a left pupil filtered signal, and a right pupil filtered signal. And as you can see, the left pupil signal is pretty consistent, and you don't see many large variations, but for the right pupil size, you can see these very large deviations. All the crosses that you can see, I hope you can see those, they're quite small. That's all the discarded data. And all the circles are the valid samples that are taken into account in the filtered signal. So that's why it's still important once you've applied all the filters to still manually inspect, because it can miss issues like this. So, and once you've gone through all the files, you can export the data. You can export it to TSV, CSV. You can resample it. And then you can continue with stuff like baseline corrections and your further analyses. So now I'm going to be showing you what it would actually look like in the PhysioData Toolbox. All right. So I've already imported all the files because that can take a while to load. So let's see. Let's first view the raw data. Okay. So this is from female orangutan Amos. And here is the raw data of the left pupil and the right pupil. You can zoom in specific sections if you want. And here's a closer look. You can see that there are some areas, some raw data samples that are probably not an accurate reflection of the actual pupil size. This is a bit of normal expected noise, especially for non-human primate, that data is naturally a bit noisier. And if we go to the next orangutan or the next session, you can see here this would be an example of very noisy data. Here the left pupil indeed is not captured accurately in this part. Later on the file here it gets a bit better. But in the beginning especially, it's very noisy data. And it is good to already look at this before you apply the filters, because you can already tell, okay, I should really take a close look, especially to the ones with noisy data. What you can also do is define, when you filter the data, is the time windows of interest. This is the whole entire time that the eye tracker was on, but of course you're probably mostly interested in the response during trials. So automatically it will detect your markers or the name of your images or your trial name, and it will create this event table. And based on this event table, you can create these time windows of interest. So this is what the raw data looks like in the PhysioData Toolbox. Then we apply, or we create a filter. As you can see, there are also other analyzers that you can use for other types of physiological data, but we're looking at pupil diameter for now. So these are all the settings I talked to you before, and these are all the standard settings are already there, and these are the ones from the guidelines of the pre-processing of the pupil size. So I typically leave these as is, but you can adjust them manually to your preferences. So here at the top, we can create these time windows of interest. So let's create one real quick. All right, automatically a time window, or epoch is what it's called in the toolbox, is created for the whole signal. But let's say I'm interested in the fixation, because I later maybe want to use that for my baseline correction. So I create this time window and it's called fixation, and then I look at my marker table and I see what this is called. So the fixation in this case is called start primer frank, and it ends at end primer frank. So I will mention here start primer frank and end value. So it should stop making this time window of interest when they see the value end primer frank. And I want this to happen every time. I want this file to be created every time they see the start primer frank. So every time until the last time they see start and end primer frank, a time window of interest will be created. So once you've done this, you can click on generate epochs, and here you can see these time windows have been created. So this epoch creation uses regex, so you can create quite complicated names. So you can use start, let me see if I can, start underscore def m, something like this. Let's see if that works. Then ends, or I can make it easier. I can probably do start underscore, let's see, save start underscore 34. If I want to look at just this trial, I will start 34 and we'll do end 34. And this will only occur one time, so I can just do one time. And there is our start 34 trial isolated. You can create more complicated, where you can just take all the trials that you want from a specific. So you can say, I want trials 1 to 10. Then you can say start value can be 1 to 10. But with regular expression, you can create all kinds of complicated values for your epochs. But for now, let's keep it at this. So I click on OK, click on OK here. And now we have the analyzer already, and we apply it to the PhysioData files. So here you see again a summary of all the filters and your time windows of interest that it uses. And you click on Apply Files, and now you can see it has been applied to all of the data. So if I click on View Analyses, so there you go. So this is what the filtered signal looks like in this data set. So you have the left pupil filtered, the right pupil filtered, and the mean pupil filtered. And now we can also do, again, a visual inspection, and you should. What's good about having this visualization also is that now we have this table epoch, and we can now see when all our time windows of interest are present. So we only need to look at what the data looks like during our time windows of interest, because the other data we don't necessarily need to further inspect if we only want to continue with our data of interest. So let's say you see a mistake. If I zoom in, for example, here you can see this dip of the left pupil. You might say, okay, I think this is unnatural. I think this is probably a mistake for the eye tracker. So I want to filter this data out. So you can use a left mouse click. And here you can choose for the left and the right eye separately if you want to accept or reject the data sample in there. So I'm going to choose force rejection sample. And then you can see the left filtered signal is gone in this spot. And it has also affected the mean pupil signal here. So this is what a regular noisy data sample would look like. It looks pretty okay. But then we look at our very noisy data. And here you can see some very unnatural results from this filtered signal. So here you would probably exclude more. But let's say you see a part of the data samples that you think is wrongfully rejected. So let's say you see these samples here and you think these are valid, not necessarily in this instance, but just using it as an example. You can also select this part and you can click on Force Acceptance of these samples in the selected range. So now you can see the left pupil filtered size is also now accounted for. That even though it was projected automatically by the filters, you can still add these parts. So once you've gone through your file, you can click if you want that you accept this, and then it's an easy way for you to keep track of where you are in your pre-processing stage. So now you can see this green one, that's the one I've clicked on accepted. And this is actually the part of the PhysioData Toolbox. Now you're ready to export your data, and you can click on Resampled Epoch Signals. So remember, it was automatically upsampled to 1000 Hertz. You can choose different sampling rates here again. And the only export that will happen is of your time windows of interest. Of course, if you leave your time window of interest of the full signal that's automatically there, then you will still have the full data set that's filtered. But you can also choose to just look at your trials or your other windows of interest. So it can export to different types of files, TSV, CSV, and Excel. And I also have an example. So this, for example, a trial export of Watana, another orangutan. And here you can see some standard names. You can see the specific actual value of your event marker. So you have start 53M and value of end 53M. And then at the end, you can see the left filtered pupil, the right filtered pupil, and the mean of both pupils. So this is the export that you will get from the PhysioData Toolbox. And if you're interested in the results of this study, so they do indeed have a larger pupil size for the orangutans with bigger cheek pads, but we also see that this pupil size is even bigger for their caretaker Frank compared to other orangutans. So yeah, thanks. This was my demo.
Awesome. Thank you so much. Thank all of you in the audience for your patience. I'm so glad that we got to see the demo, Anouschka. Sylvain, we would love to have you come back. We can get to the Q&A. We are running a little bit over on time, so we're going to get to as many questions as we can. We've gotten at least 50 questions in the chat. So thank you all so much for your engagement and your attention. As a reminder, this is being recorded. It will be shared with everybody later this week. The first thing we're going to do is a couple of quick polls for the audience. We would love to know, have you used eye tracking in your research? Are you using it now? Have you used it in the past but not now? Or haven't used eye tracking yet? Just a little bit of engagement to see, do we have some current customers here? It seems like we do, but also some people who are maybe very new to it and interested in using eye tracking and trying to learn. Okay, I'm going to give it a couple more seconds. So we have one more poll. Okay, I'm going to end and share the results. So most of you right now are currently using eye tracking. Some of you are not using it now but have in the past, and then about a third are not yet. So one more question, curious about your field of research. We have to keep it kind of brief, but where's your field of research? If you are not represented in this list, we'd love to hear about it in the chat. What field of research you're in, and especially how it relates to eye tracking and pupillometry. Linguistics, cool, very cool. Okay, all right, I'm going to share the results here. Lots of psychology and neuroscience, few of you in medical, many of you in human factors and engineering. We do find that that's a pretty popular field, and then some other people. So awesome. Well, again, tons of questions. Thank you all so much for the engagement. I'm going to break them up into sort of themes, and anything that I don't get to, I'm going to do a write-up, and we'll make sure that it's available for everybody who's interested in the questions that were asked today. OSC Sports Science, R&D, very cool. So there was a couple questions about the Tobii systems. I can answer those quickly. The Tobii Pro Spark does provide estimates of pupil diameter, but it's a single camera system. So unless you have the participant in a chin rest or head fixed, you may find the effect of Z distance as artificially increasing the pupil diameter. So if they get closer to the screen, the pupil diameter might look larger. That's why we recommend a 2-camera system. But you can use the Spark again if you kind of take those things into consideration. Someone asked earlier about using the Fusion 250 with infants. That should work just fine for pupil diameter. It's a dual camera system. Somebody asked about Tobii Pro Lab for pupil data. You would export the pupil data. None of the analysis can happen in Pro Lab, but you export the data and then do all of your pre-processing and further analysis outside of Pro Lab. Okay, so for the two of you, lots of questions about practical issues. How to find the baseline, do you need a jitter, minimum trial duration to see a pupil area response, lots of questions about isoluminance and best practices, and controlling lighting. So Sylvain, I'd love to hear a little bit about some, I know these are huge topics, we have like 5 minutes left, but any advice that you can give, and we can provide a more detailed answer later, but advice on those questions.
It's going to be, from my point of view, quite related to the specifics of your study. In infant studies, for example, when the baby is willing to participate, you tend to throw all the trials as quickly as you can to get as much data as possible. So like a pre-trial baseline can be complicated because it follows from the previous trial, and who knows what you're dealing with as far as arousal, etc., carrying over from the previous trial. As much as possible, you do want a period before any stimulus or event of interest, so you can get a baseline. But usually a baseline of 100 milliseconds will be enough. In the case of the oddball data I showed today, I used 6 samples. I was sampling at 60 hertz. So now I used 3 samples, so 50 millisecond baseline just to line up the data. But obviously, the thing you want to do more than anything is go after robust effects that are going to be tolerant to noise, because you throw in a lot of noise in these tasks.
Great. Anouschka, do you have any advice on this hefty topic of setting up a study properly? Oh, you're muted.
Sorry, I unmuted my laptop instead of my phone. Of course, the typical control for luminance in your trials. That's one advice. Keep your lighting stable. But yeah, that also depends. If you use a Tobii glasses, for example, that might be more difficult. And I do use the Tobii glasses also for pupil diameter. But yeah, try to take reliable baseline measures. But yeah, it's noisy data inherently. So it's influenced by so many factors. Most of them we can't control. Some of them we can control for. So indeed, robust effects are best.
Great. So we actually did have some questions about real world eye tracking. So if you've used the Tobii glasses, I'd love to hear. People are interested in advice on how to study changes in pupil diameter in outdoor or real world tasks where the environment maybe can't be real controlled. Imagine like human factors or like flight simulator applications. What advice do you have?
So in my setting, I do try to control for most. So we have stable lighting, curtains closed, all that standard start stuff as a first start. And then we take frequent baselines. So we have, again, fixation cross. Even in real life settings, we also use those, of course. But for flight simulations, I think trying to keep your luminance, contrast, intensity as stable as you can. And I think there are also some packages that will show you what your luminance, intensity, contrast is as a whole. But that's, of course, more easily detected during actual simulations and not in a real world setting. So I am not sure if there's a good way to control for those. I am lucky to be in a fairly controlled environment still, unfortunately. Yeah.
And Sylvain, I don't know if have you done any pupillometry with non-screen based eye tracking or have any advice?
No, but it is a question I asked in the neuroscience course that students take. At the end of the day, they need to, provide a little video answer to a question I asked them. I asked them why they shouldn't do eye tracking outdoors if they want to do pupillometry. Because there's all this noise that will get in the way. But obviously, with something like glasses, where you do have a feed of the environment, figure out what they're looking at, you could conceivably, I don't do it, but you could conceivably sort of derive some measure of scene luminance frame by frame and use that as a potential reference to sort of control pupil diameter. Filter it, and then see what's left.
Okay, great. And I know you provided all the resources and the materials from your presentation. And Anouschka, how can people who are interested in using this toolbox find it?
I can send the link in the chats.
Okay, great.
Maybe right now. Let me see if that would. Did I send it to everyone?
Yes,
Okay,
I see it at least. I hope everybody does. We'll make sure that that's in the follow-up as well. So if you have a data set from a pupil, or from a Tobii system, there is pupil diameter data in there, and you can use that to practice with the toolbox. If you do not have an eye tracker and you're interested in trying this out, we have sample projects on our website, our Tobii Connect portal, connect.tobii.com. You can download a project, download the pupil diameter files, and use it with the toolbox without having to purchase anything to test it out. So many questions, lots and lots of questions, but thank you so much for your time. I guess my final question to you all is, what are you excited about for the future of eye tracking or pupillometry research, or in your fields, or what sort of your dream features and tools that you would like to see? And Anouschka, let's start with you.
I would love to move more towards a real life application. So if we can solve the outdoor issue, that would be great. I mean, it is somewhat possible, but it's not perfect yet. And also probably more complex integration of multiple participants into one data set with preferably, I would love to study groups with 4 people with the Tobii glasses and have markers and have different physiological measures all integrated. So that's what I would be very excited about to move towards. And I think that we're really getting there. So
yes,
that would be
awesome.
very nice.
Awesome. Yeah, how about you, Sylvain?
Well, I hope it doesn't move too fast because I'll be out of work. It's a great time right now because the technology is great, but there's still much to explore. I did promise today to talk about acceleration, which I find really, really interesting, but in the interest of time, I didn't dive into that today, and I still need to do some work. But I do think that perhaps the rate of change of pupil diameter can be an index of the information processing clock, which is key to a host of cognitive abilities. And it's relevant for me with infants, but it's relevant for adults and also aging. And I think looking at second order properties of and changes in pupil diameter could be informative with respect to fundamental individual characteristics of information processing. So that's something I'm keen to look into.
Yeah, and I think there's a lot of space there for, because people are, measuring changes from baseline, but how fast the change is occurring is super interesting. I totally agree with you there. So thank you so much. We are definitely over time now, but thank you both so much, and to Dr. Kret, who had to leave a little bit early, for your presentation, for being here with us, for teaching us, and thank you all in the audience for participating. Again, I'm going to get to all these questions and put together a document that can be emailed out and can also hopefully be downloaded, so that way we can get to all the specifics of the questions that we weren't able to get to today. But thank you for your engagement. We hope to see you in another webinar. Oh, also thank you to the Tobii team who helped put this together, all of our marketing team. We could not do this without you. Thank you, Valentina. Hope to see you at another webinar again soon. Feel free to email me if you have any other questions. Have a great day.